Let's kick off this explainer by imagining the human cell not just as some microscopic blob of biology, but as a bustling, incredibly advanced manufacturing plant. And like any massive factory, it relies on a highly guarded master instruction manual to keep the whole operation running smoothly. So today, we're going to unpack exactly how this microscopic factory operates, how it stores its immense amount of data, and how it actually reads that data to quite literally build you from the ground up.
So, what acts as the master control center for every single trait you possess? Well, the answer to that points us straight to the very heart of the cell. If we zoom into our biological factory, we arrive at the nucleus. Think of this as the ultimate executive office. It's a double-membrane command center that dictates absolutely everything. It has these specialized pores that carefully control the entry and exit of vital materials like RNA and proteins, and it houses the genetic material that controls all cellular processes.
But, as you probably know, biology loves a good plot twist. While the nucleus is the master control center for virtually all of our cells, our red blood cells and platelets somehow manage to function completely without this master blueprint. They literally don't have a nucleus at all. It's this amazing evolutionary adaptation that basically clears up space so red blood cells can carry a lot more oxygen, even if it means sacrificing their own internal command center.
Now, we didn't just figure all this out overnight. Our understanding of this fascinating structure really built up over time. It started way back in 1831 when Robert Brown first discovered the nucleus of the cell. Sometime later, a scientist named Altman identified the actual substance inside, calling it nucleic acid. But the real game changer, the breakthrough that cracked the code of life, hit in 1953 when Watson and Crick put forward their famous double helix model of nucleic acid.
Okay, moving right along into our first main section, the master code, decoding DNA and RNA.
Inside that executive office of the nucleus, the instructions are written in the chemical language of nucleic acids. The alphabet of this language starts with nitrogen bases, specifically adenine, guanine, cytosine, thymine, and uracil. When one of these nitrogen bases bonds with a sugar molecule, it forms what we call a nucleoside. But to get the ultimate building block of our genetic code, you have to add a phosphate group to that mix. That specific combination, a nitrogen base plus a sugar plus a phosphate, creates one single nucleotide.
These building blocks perfectly highlight the key differences between our two main chains of nucleotides, RNA and DNA. RNA, or ribonucleic acid, is a single helix structure featuring a ribose sugar and that unique nitrogen base, uracil. DNA, or deoxyribonucleic acid, is the famous double helix structure with a deoxyribose sugar and the base thymine. Now, generally speaking, RNA is not the main genetic material, but there's a big exception here. In specific viruses like HIV, polio, hepatitis A and E, and corona, RNA actually does step up to act as the primary genetic material.
If we look closely at how these molecular chains physically fit together, it's pretty incredible. Nucleotides from the same strand connect to each other via really strong phosphodiester bonds to form the backbone. The phosphate group attaches to the fifth carbon of the sugar, while the nitrogen base hooks onto the first carbon. When you have two strands of DNA, they actually run in opposite directions. We call this an anti-parallel direction, flowing from 5' to 3'. So even though they're flowing in opposite directions like a two-lane highway, the nucleotides remain perfectly equidistant from one another the whole way down.
But how do those opposite strands actually know who to pair up with? Enter Chargaff's rule. You can think of this as strict biological matchmaking. The rule dictates that the number of double-ring structures, which are called purines, absolutely must equal the number of single-ring structures, called pyrimidines. Specifically, the purines, adenine and guanine, must equal the pyrimidines, cytosine and thymine. It's this incredibly elegant, perfect mathematical equation hiding right inside your cells.
Let's jump into our next section, packaging the blueprint, the spatial problem.
2 meters. That number is just staggering. If you were to take the DNA out of just one single eukaryotic cell in your body and stretch it out end to end, it would be exactly 2 meters long. And yet, it's unbelievably thin, only 2 nanometers wide.
Seriously, think about that for a second. How is it physically possible to pack 2 full meters of material into a space so impossibly tiny that you can't even see it without a powerful microscope? It's literally like trying to fit miles of fishing line into a thimble without tangling it up.
Well, the cell solves this massive spatial problem by using specialized spools. That 2-meter-long DNA wraps itself super tightly around structural proteins called histones.
This combination of DNA and protein is what we call chromatin. When the cell gets ready to divide, this chromatin condenses into incredibly solid, tight structures known as chromosomes. And the specific functional groups along this tightly packed DNA, those are what we call genes.
Inside human cells, we have exactly 23 pairs of these highly condensed genetic filing cabinets, making 46 chromosomes in total. The first 22 pairs are called autosomes, and they handle your non-sex characteristics. The 23rd pair are the allosomes, the sex chromosomes, where females have XX and males have XY. What's really fascinating here is that the female X chromosome also carries instructions that determine some completely non-sexual characteristics too, things like color vision, blood clotting, and even aspects of brain development.
Which brings us to our next part. When the code glitches, chromosomal aberrations.
When a factory is dealing with millions of precise microscopic structures, occasionally tiny deviations happen in the exact number of chromosomes. These deviations on the 23rd pair lead to specific syndromes. For instance, Turner syndrome is a monosomy, meaning there's only a single X chromosome with no pair. This occurs in females and is characterized by short stature and infertility. On the flip side, trisomies, where there's an extra chromosome, create totally different results. Super male syndrome has an XXY makeup, resulting in males who are taller than average, often with criminal tendencies. Super female syndrome, an XXX makeup, results in taller females with normal fertility. And Klinefelter syndrome is an XXY makeup, resulting in males who exhibit feminine characteristics and are infertile.
Now, these glitches aren't just limited to the sex chromosomes. They can absolutely happen on the autosomes as well. Down syndrome is a prime example of an autosomal aberration. It's a trisomy occurring specifically on the 21st pair, which brings the total chromosome count to 47. Because it happens on an autosome, it can affect either gender and is characterized by specific physical traits and developmental patterns.
All right, let's head into our final section, the assembly line, protein synthesis. At the end of the day, building a human being all comes down to proteins. And proteins are formed by connecting amino acids together with peptide bonds. There are exactly 20 amino acids that form the proteins in the human body. 10 of these are non-essential, meaning your body functions as its own internal factory to just produce them on demand. The other 10 are essential amino acids, meaning you literally have to get them externally through your diet. Together, anywhere from 100 to 3,000 of these amino acid units will link up to form just a single chain of protein.
But remember, the DNA never actually leaves that executive office in the nucleus to build these proteins. Instead, it relies on a dedicated 3-part workforce of non-genetic RNA. First up, you have mRNA, or messenger RNA. Its job is to act like a scribe and copy the specific instructions right from the DNA. Next is RNA, or ribosomal RNA, which stays out on the factory floor to make sure the workbenches, the ribosomes, are totally prepped and ready for assembly. Finally, tRNA, or transfer RNA, acts as the transport crew, carrying the exact amino acids needed from the cytoplasm over to the ribosomes.
These 3 types of RNA pull off an exact 3-step assembly line that is happening inside you right this very second. Step 1 is transcription. The DNA's symbolic language, called a codogen, is copied down by the mRNA into triplet codons, basically 3 nitrogen bases for every one specific amino acid. Step 2 is translocation. The tRNA reads this mRNA using its own complementary code, an anticodon, and transfers the correct amino acid over to the ribosome. Finally, step 3 is translation. The ribosome takes those delivered amino acids, clicks them together, and actually builds the final protein. This continuous, sequential flow of biological information, starting from DNA, transferring over to RNA, and ultimately manifesting as a physical protein, is the absolute bedrock of our physical existence. It's so fundamental to life that scientists literally call it the central dogma of molecular biology. It is the one unbreakable rule of the biological factor. I want to leave you with this thought. We've just seen how 2 meters of instructions are perfectly spooled, copied, transferred, and translated just to build a single protein. When you realize that a tiny variation on just one of those 46 microscopic spools can fundamentally alter a person's entire physical and mental development, it totally puts the sheer precision of the human body into perspective. It makes you wonder just how incredibly precise and yet fragile the instruction manual making you who you are truly is. And it really makes you think about what other incredible mysteries are still buzzing away inside your own biological factory right now.